Welcome, welcome, welcome. We're now on part 3 of our 5-part series of ALK 101. Like I said before, this is like a 3-year process of trying to get this put together and putting together the caretakers and ALK patients, I guess you can call us. And these are the people behind the scenes who are making things happen for us. And I want to thank Dr. Colin Barton, who will be presenting tonight, Dr. Ellie Urban, who presented her first series, Professor Mark Muskovich, and Emily Van Nancy will be on our last part 6. Jeff Sturm was the last week, part 2, and Dr. Ken Kelver, who I believe is in the crowd somewhere, who also chimes in. So welcome everyone, and I'm going to show one thing real quick before we do a polling. Well, you want me to do a polling first. So first question will be length of diagnosis. All right, I'm gonna end the poll. Leading the pack is 1 to 2 years at 41%. We got 19% 1 year or under, 19% of 3 or 4 years, 5 to 6 years at 9%, 7, 8, 2%, 8 to 9, 2%, and we have 7% of 9 to 10. Stop sharing that. And you might ask, why am I reading these out loud? Because our recording does not pick them up. So let me go with the next one. Okay, are you currently a volunteer for ALK Positive? Okay, and the poll. All right, 9 out of 60 people, which is 15%, say yes. 85% says no. So 85% of us need to get more involved. Stop sharing that and go back. I got one last question. And the question is, do you prefer that question mark to be involved in decisions about your treatment? Do you prefer to leave it entirely to your doctors? I tend to consult with my ALK community. All right, I'm gonna end the poll, share the results. All right, so you guys see that? Yes, no? No? All right. All right, 89% of us leave the decisions or treatments that they want to be involved in the treatment. 2% leave it to the doctors, and 9%, 5 out of 55, which I thought would be higher, consult with other ALKs. So those are my questions, and basically, I don't really have a prepared speech for Colin because I figured, what can I say about Colin, right? If there was like a Mount Rushmore of ALK Positive members who made ALK Positive who they are today, he would be ahead of one of the heads. I wouldn't be ahead. Summer wouldn't be ahead, but Colin would be the head. So I'm going to leave that up to let Summer make a couple of statements real quick, and then Summer, do me a favor, pass it back to me. I got to present something.
Mark, I don't even know how to pass it to you. Sorry.
With your sparkling personality, that's wow.
Oh, pass it back to you. Okay, gotcha. I thought, okay, we're good. Okay, thank you all so much for joining us on part 3 of our 5 ALK 101 series. This has been, like I've said, each week. This has been a long time coming. The ALK Talk team has been brainstorming ALK 101 for years, whether it was a video series or a welcome guide or some type of handout. We knew that there was a need for all of us not to be kind of lost and floundering upon diagnosis. So we talked to many people along the way, trying to figure out just exactly what the special sauce was going to be for this series, what needed to be included. And we've made adaptations along the way. As you know, it was supposed to be a 4-part series, and it recently switched to 5. But finally, what got the ball moving was we sat down with Dr. Ken Culver, the ALK Positive Inc. Director of Research, and things started to fall into place, and we had more meetings and got our team assembled. But the point that I want to make is all along, all this time, we knew that we wanted, or I should say that we needed, Colin Barton involved in this project. It wasn't going to be a project without him. So one way or another, while we're coming up with our ideas, we knew we were going to have to convince him to join along. Because once you join the ALK Positive support group, or let's say that's not your thing and you go through the YouTube videos, you realize what a gem he is in our ALK Positive family. He's our voice of research. He is our voice of reason. But what a lot of people don't know is that all of this science that goes on behind the scenes of ALK Positive would not be possible without the foundation that Colin laid. The research library, multiple medical committees that I can't even keep track of, meetings with biotechs, meetings with KOLs, clinical trial committees, and spreadsheets, and all of that was all came to fruition because of what Colin started. So I have to say, like I am heartbroken for all of us that we're even brought together on Zoom today, and I'm heartbroken for Colin that his well-deserved retirement from a fruitful career was altered. I am so grateful every single day that Colin's on my team, that he's on all of your team, working for us, making the magic happen while we are sleeping. So thank you, Colin, for being here with us tonight. And Mark, am I handing it back to you?
Yeah, sure, real quick. I'm just gonna go real quick through one thing. So, okay, I'm sharing with you our ALK Positive website. And when you go to ALK Talk, which is up here, and you scroll down, this is how you get to everything for ALK Talk. This is our calendar. And I want to tell you, this week coming up, we have a big week. Tomorrow at 1 o'clock, Dana Carita will be hosting the Death Cafe, yoga with Hanka. We get ALK Chat at 1 o'clock, guided imagery at 3, art therapy at 1 o'clock, and then Heather Smith will be presenting also. And then we have our men's group at 9 o'clock Eastern time. So this is how you get to the calendar. As simple as you push in this, and it'll take you right to the Zoom room. So, and also back up to ALK Talk. You can get to the our past ALK Talks, which you can check out the most recent videos or our archaic videos. So anyways, that's the easiest way to get a hold of us. And you also can contact us with any ideas or input, and we would love it. So Colin, take it away. Show's yours.
All right, well, thanks very much to Mark and Summer for putting together all the ALK Talk things. It's just absolutely incredible asset to the ALK community. And thanks for inviting me to speak. I'm not a natural speaker, so it's hard for me. But Summer, thanks for the kind words. And I'll be sharing a PowerPoint presentation. The PowerPoint presentation will be available as a link later, so you'll be able to actually download the PDF in the future if you wish to. So I'm going to share my screen here. But firstly, just like to, before I do that, just welcome everyone. This was a real challenging presentation to do. Originally, Mark and Summer asked me to do a presentation about what to do when you get cancer progression. And I have a post in the ALK Positive support group that gets widely referred to. And they said, just try to put that into a video. And as I started doing that presentation, I thought to myself, I'm going to be using all these terms that people that are new to the cancer world don't understand. They don't know what they mean. And I remember when I joined the support group back in 2017, early 2017, and people would say, I'm Ned. And I was like, who's Ned? And they'd say ALK. And I was like, I didn't even know what ALK stood for. I'd sort of read it, I'd remember it, and then I'd forget it 2 weeks later. And, trying to learn what all the different TKIs were and all that sort of thing. So it's a huge learning curve. And so as I put together this presentation, I realized there was no way I could cover in 30 or 45 minutes kind of the vocabulary and what to do if you have progression. So I decided to split it into 2 presentations. And so today's presentation is basically just understanding the ALK language and the cancer language and a lot of the things that you read about on the support group. So terminology I'm referring to is basically derived from what I see on the support group that people seem to not understand when they're fairly new to the cancer world. So, if you've been following the support group for 2 or 3 or 4 years, none of this is going to be new and you're going to pretty much know most of this, but this is really oriented more towards the people that are pretty new to the cancer world and need that background so that next week you'll understand the presentation. And I do hope that you all reviewed the fantastic presentations that Ellie and Jeff did. I know that my presentation won't be nearly as good as what either of them did, but yeah, they did absolutely fantastic presentations. So I'm going to share my screen and hopefully everything will work well. Today's presentation is building on parts 1 and part 2. So part 1 was by Ellie about I've just been diagnosed with ALK cancer, what now what? And part 2 was by Jeff, building and managing your care team. Today's presentation will be about learning and how to learn and how to understand all the terminology that's necessary to make informed decisions. And it was an interesting poll, and the vast majority of you do want to be involved in making your decisions about your care. So that's a great start. There'll be 2 parts. Firstly will be the presentation, and then afterwards we'll have questions and answers. And Ken will be helping me with that. Today's ALK talk will be recorded and it'll be uploaded to the YouTube channel, and there'll be a PDF available, and the PDF will show on the YouTube channel, so you can link it from there. So just a little bit about myself, super briefly. I'm a patient, just like most of the rest of you. I was diagnosed in 2016. In fact, this month is my 7-year anniversary. So I'm very excited about that. I can tell you back in 2016, there were very, 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 very few people that had survived 7 years. I think literally in the support group, when I joined in 2017, there might have been 2 people that had been survivors for 7 years. So it's a very different world now for people that are getting diagnosed, because you've got all these other people that you can see that have survived for 6, 7, 8, 9, 10 plus years. But when I was diagnosed, you really didn't think you were going to be around for very long at all. So I'm thrilled to be here and thrilled to be feeling well and thrilled to be able to share my experiences with you. In my career, I was a dentist all my life. I grew up in Australia, practiced half my career in Australia and then half my career in the US. And I retired in 2015 and thought I'd be doing what retired people do, and then I got diagnosed the year after, which was definitely not part of the retirement plan. I've been a volunteer for the organisation ALK Positive Inc. And it's been super rewarding, I've got to tell you. Not only helping people, but just the people that I've been working with are all very positive. You know, you look at Mark or Summer or Kirk or all the people that are with the organisation, they're all super, super positive people and a wonderful group of people to be working with. I was chair of the medical committees from the time it was first founded in 2017 and continued as chair until Ken Culver took over that role towards the end of last year. And I was also a board member of ALK Positive. It was actually called something different back in 2017, but evolved into the non-profit organisation and eventually became a registered non-profit organisation. And I resigned from that towards the end of last year also. And I am still currently an active member of all the medical committees. So I'm still very, very involved with volunteering. And as most of you would realise, I am a very regular contributor to the Facebook support group page, and I learn a lot by reading people's posts and the comments. It's very informative reading the comments. There's a lot to learn. And Summer sent me this photo. This is from the summit last year. And we've got Mark and Summer and myself there. And as I mentioned earlier, we wouldn't be here without the great efforts that Mark and Summer have been doing. So thank you to both of them. So a little bit of an overview of today's presentation. Firstly, we'll just be doing an introduction, what we're trying to achieve with today's presentation and talking about what personalised or precision medicine is and why it's so important. And then we'll be getting into some terminology, firstly about just basic cell terminology and then cancer terminology, and then measuring lesions and then measuring progression and response and then cancer staging. And then we'll be getting into discussion about progression. Now, today's talk is not about what to do if you have progression. Today's just about learning all the terminology and getting the background for next week's presentation. So the goal for today is to give you, and today and next visit, the next presentation next week. The goal for these two presentations is to learn what to do as our patients and caregivers if cancer progression is suspected. And why do we want to learn what to do? Because as Jeff mentioned last week, there really is a better chance of living longer and having a better quality of life if you're well informed and can be involved with the decision. I was a dentist, and as a dentist, I know, working with dentists myself, doctors are busy and doctors are people, and nobody can spend the time that you're going to spend researching your own healthcare. You might like to think that the doctor can, but doctors are people. You know, maybe their baby was up crying all night. Maybe their wife's been giving them a hard time. Maybe their kid got into trouble at school. Maybe they've got their own health problems. Who knows what's going on in their life? But, you know, doctors are humans. They make mistakes and they're not always fully attentive. And they're certainly busy rushing from one patient to the next and don't necessarily have the amount of time that we would like for them to provide us with. So being well informed and understanding your disease is super important to doing the right things with your treatment. Pretty much all the questions and topics that I'm discussing are based on things that I see in the worldwide support group. The information that I'm providing is from reputable sources such as the National Institute for Health, NCI, and then the leading doctors and researchers in the ALK world. As Ellie mentioned, we're not providing medical advice, but we are trying to prepare you for what to discuss with your doctors. And we'll be referring to ALK cancer, pretty much all stages, and most patients are lung cancer patients, but this will probably apply to any other types of cancers as well. So This is Dr. Gaynor, and he was in a debate at the Targeted Therapies Conference just last month. And they were debating whether lorlatinib should be always given first line or whether it shouldn't always be given first line. And Dr. Gaynor was assigned the side that had to argue that it should not always be given first line. And his summary at the end, and I'm paraphrasing a little bit, but it was very close to this, was the essence of precision, which is sometimes known as personalized medicine, is that we should not take a one-size-fits-all approach, and that informed decisions should be made between patients and clinicians, considering all the factors. And why? Why is that so important? Because every patient is different. We've all got different health. We've all got different priorities in life. Some people have young kids and their goals to stick around to see their kids grow up. Some people have grandkids and they want to see their grandkids grow up. Other people, they don't have kids, they're retired, and they just want a high quality of life, and maybe they would be prepared to sacrifice a little bit less time and have a better quality of life. So every patient is different, but also every cancer is different. There are no two cancers that are absolutely identical. And so we shouldn't all be treated the same. Even if two people had exactly the same cancer, they would be treated different. Or if you had exactly the same cancer, but or exactly the same patient, they could have two different cancers. And to make an informed decision, you need to understand what's being discussed and be able to communicate that. And that's where today's presentation will help you. And you need to make sure that all the relevant factors are discussed, and that's what we'll be talking about at the presentation next week. And it is your life. Be empowered. As I mentioned, doctors are people. And so you need to be able to kind of know when you need to keep them on track and get them focused. Now, this is something I wasn't going to present, but Jeff presented it last week and I thought it was so good I've copied it. He said, learn as much as you can continually, do your own research, visit and learn from the ALK Positive Facebook patient support group, study the ALK Positive website. And I'm going to tell you there's a huge amount of information there. If you really click into all the tabs and really explore them, you can learn an incredible amount from the information that's on the website. Attend the ALK talks and review the videos. Ask questions all the time. And I added this one, learn how to use Google. Now, when you use Google, make sure that you click onto items that are from a reputable and reliable source, but you can learn, everything I've learned is from everything that you see here. I was a dentist, I wasn't an oncologist. People think I know all about ALK. I don't know all about ALK. I'm just learning all the time. And then, as Jeff said, be ready when the moment comes. Progression is going to come. It's not a matter of if, it's just a matter of when. So be ready. And then when you're ready, you'll be able to provide intelligent input to decisions once you're fully informed and the options are understood. So to be able to understand and communicate, we need to be familiar with the terminology. And as I mentioned, when I started, it was just a jumble. I didn't know what NED was or any of these other terms. All those words basically looked like this, just looked like a huge jumble of words that made no sense. But as you become more familiar with it, as you keep reading articles and reading comments and items in the support page and talking with doctors, you learn and you become more comfortable with the terminology. I'm just going to take a sip of water here. Mark, if I'm rattling on for too long or too slow, please tell me to hurry it up.
No, you're doing a great job. Keep it rolling.
All righty. So we're going to just look at this little diagram here. We're starting here with a healthy cell. And normally what happens is cells replicate and then they form healthy tissues. And tissues could be skin tissue, bone tissue, can be the parts that form around your digestive gut, can be your hair, it can be anything. And so that's what happens normally. But if somehow the inner part of that cell gets changed, it becomes something different, becomes a cancer cell. And then when the cancer cell doubles and doubles and doubles, it can become a tumor. So a cell is the smallest unit that can live on its own. It makes up all living organisms and the tissues of the body. It has three parts. It has the nucleus, which is this center part, which is where the DNA is. And the DNA is the part that allows cells to replicate. And it's a miracle that they ever replicate correctly. It's even more of a miracle that they replicate correctly nearly all of the time. But they actually make mistakes by the millions and billions every day. But most of those mistakes don't matter or get cleaned up by our immune system. The cytoplasm is the rest of the cell. It's basically all the engine that keeps it all running. And then the cell membrane is around the edge of the cell, and it controls what goes in and out of the cell. That's important to know. So a tissue is a group of cells that have similar structure and function together as a unit, as I mentioned, like skin or bone. A lesion is an area of abnormal tissue. So that's an important term to remember. A lesion is an area of abnormal tissue. So if you get a cancer cell and it starts replicating, it can form a tumor. And most of us have what's called a solid tumor, which means it does not contain cysts or liquid areas. And this is different to the other sort of tumors that are in the blood and so on. So malignant cancer can grow and spread to other parts of the body, whereas a benign tumor is not a cancer, and it can grow, but it can't spread. And so, unfortunately, our type of cancer is malignant. It can grow and it can spread. But there's different types of solid tumors. So most of us have carcinomas in the ALK world, but not all of us. Carcinomas are ones that are formed by the cells that cover internal organs. And then the most common type of ALK carcinoma is adenocarcinoma, but there are many other types as well. I'm actually squamous myself. It's only about 1 or 2% that are squamous, but I'm actually squamous. And then some of the patients in our group actually do not have carcinoma, they have sarcoma. And so when you see that term, when someone introduces themselves and says they have a sarcoma, that's a little different. So a mutation is any change in the DNA sequence of a cell. It's not necessarily a bad thing. It can be harmful, but it can be beneficial, or it can have no effect at all. And mutations can be caused by mistakes during cell division. They can also be caused by exposure to DNA damaging agents in the environment. At this stage, there's no evidence that that's the case with ALK. It may turn out to be the case, but at this stage, there is no evidence that that's the case. So it seems at this stage that it's just totally related to mistakes made during cell division. And then, as I mentioned, cancer can spread, and that's called metastases. And again, people would refer to METs, and I was like, what's a MET? So a MET is just an abbreviation for metastasis. It's the spread of cancer cells from the place where they first formed. And where they first formed is called the primary tumor, and then they can spread to another part of the body. The cancer cells break away from the original part, the primary tumor, and then they travel through the blood or the lymph system, and they form a new tumor in other organs or tissues of the body. What's important to know here is that a tumor is always designated by the type of tissue that it originated from. So if you've got a lung cancer and it spreads to your brain and you've got a tumor in your brain, that's not called a brain tumor. It's still lung cancer in the brain. Or if it spreads to your bone, it's not bone cancer. it's still lung cancer in the bone. And so, as I said, the new metastatic tumor is the same type of cancer as the primary tumor. So what's very important for a lot of reasons is being able to actually measure how we're responding to the treatment. And there's actually been a very complex, and we won't get into it very much, but a very complex formula for figuring out how we're all responding to the treatments. And it's called RECIST. It stands for Response Evaluation Criteria in Solid Tumors. And if you look at this little graphic here, it talks about complete response, partial response, stable disease, and progressive disease. And we're going to, on the next slide, see what those really mean. But in order for us to do that, we've got to have a tumor that can be measured, and it's measured via imaging. What do we mean by imaging? We mean things like PET scans and CT scans. And it measures the response to the treatment. And I'm going to skip through this really quickly, but it'll be on the PDF for you to look at in more detail later. But a measurable lesion basically has to be at least 10 millimeters to be measurable. So if you've got a 5 millimeter lesion, that is not something that can be used as a measurable lesion. And target lesions are all the measurable lesions representative of all the organs where the cancer might be. So whether it's in the brain or in the bone or in the lung, wherever it is, they're all measurable lesions. And they're all recorded. And then there are other types of lesions which are called non-measurable lesions. And that includes lesions in the bone because it's really hard or impossible to measure lesions in the bone. Also doesn't measure things like leptomeningeal disease, pleural effusions, things like that, but they are still part of the whole RECIST standard. So you've probably all heard about complete response, which is sometimes also known as no evidence of disease, which is sometimes known as NED. And basically a complete response means that on the scan, you can't see any evidence of the cancer there. All the cancer has disappeared on the scan. However, what's really important to understand is that a scan cannot detect a tumor, and I'm probably being very conservative in this estimate, 100,000 to 1 million cells. It could easily be 5 million or 10 million before it's detectable. So just because you don't see anything on the scan doesn't mean there isn't any cancer cells there. It just means that there might only be 100,000 of them. And then also, if tumor markers are being followed, and this will probably become more common in the future, I suspect, a complete response also has to be indicated by the tumor marker levels going back down to a normal level. So what about a partial response? Well, a partial response is where the target lesion diameters as a total compared to what they were at the start, not compared with what they were at the last time, but at the start, have decreased by at least 30%. So if they've decreased 20%, that doesn't count as a partial response. And then, so what's progression? Well, again, progression isn't just a little growth. So if you had a 2 centimeter diameter tumor and it increased to 2.2, that would only be a 10% increase, and that would not be considered progression. That could just be the variability because scans get read differently, they use different machines. And so to give a margin of safety, they say it's got to go up by at least 20%. Or if there's any new lesions or any distinct increases in non-measurable disease, that all counts as progression. So if you get leptomeningeal disease, or if you get new bone tumors, even if you can't measure them, that will be progression. We shouldn't get too hung up on these numbers. Clinically, in reality, there's probably no difference between someone whose tumor grew by 19% or grew by 21%. And so what is stable? Well, stable is everything in between. Now, it's not enough shrinkage to count as a partial response, but it's also not enough increase to qualify as progression or progressive disease. So stable is anywhere between a 30% decrease and a 20% increase. And I'm going to use this little diagram here, which is my own tumor size from the time I was diagnosed in 2016 until present. And when I was diagnosed, my tumors were around about 5 centimeters diameter. And I actually started on the inappropriate therapy because I didn't have the biomarker testing results back yet. And so they actually started me on immunotherapy, and you can see what happened. The tumor kept on growing very rapidly. In fact, in a period of only 6 weeks, it doubled in size. But then once I got started on the ALK TKI, I actually started on crizotinib, which was the only approved ALK, or was the most commonly approved first-line ALK TKI at the stage. The tumor size shrank rapidly, but it never went away completely. You can see I've always had a tumor ever since. It's always been about 1 centimeter diameter. And so I've been technically stable all of that time. But if we zoom in on this part and ignore the huge variations at the beginning, you can see there have been lots of little ups and downs. And it's really important to not get hung up on all those little ups and downs. This is totally normal as you get different angles and different readings and different machines. There's going to be some variations. So I'm just using this as a really good example to help people relax when they say, oh, my tumor went from 1 to 1.2. Well, you know, this happens. You don't necessarily get stressed out about that. So the complete response is the best outcome we can get, but stable disease is a very desirable outcome in aggressive or advanced cancers such as ALK. Now, what a lot of patients getting diagnosed nowadays don't realize is that they've done retrospective studies showing that without any therapy at all, if you didn't even have chemotherapy, the median survival for ALK cancer back in the early 90s was literally less than a year, probably 6 to 9 months. And so we're totally spoilt nowadays, you know, and so a stable disease is a great outcome. Metastatic cancers are rarely able to be cured, but with these new treatments that we've got, it's led to wanting to actually measure how long people survive without getting progression. And this is known as progression-free survival, or PFS, and you're going to see that used a lot when you start reading studies. It's sometimes known as time to progression. So PFS is just a new way to analyze how long a person lives with cancer before their cancer gets worse. As a result of these improved treatments, doctors are increasingly measuring success in terms of quality of life and symptom-free disease, rather than just the size of a tumor. And so this is an example of living symptom-free. This is me, and clearly having a great time. And I've got cancer in this picture. I'm not cancer-free. I'm not even no evidence of disease. I'm just stable, but I'm having a great life. So let's not get hung up on being no evidence of disease. What's really important is just to stay stable. Now you'll hear the terms signs and symptoms, and these are really important to understand the difference. A sign is something the doctor can see or perceive. So if you've got a high blood pressure, they can measure that. If you've got a lump, they can feel that. If you've got a rash, they can see that. But a symptom is something that's apparent to the patient. So if I've got pain, the doctor can't feel or see that. If I've got a lack of appetite or constipation, that's not something the doctor knows about. If I'm feeling really tired, that's not something the doctor knows. They're all symptoms. And so the main thing is to be able to lead a symptom-free disease, live with a symptom-free disease. So we've all heard about cancer staging. You might have heard about the term local or early stage. And all the experts agree that local early stage includes stages 0, 1, and 2. Stage 3, which is divided into 3A, B, and C, some people will call it early stage, some people don't. Some people will call it late stage. So it's kind of it's a gray area. And also, I think there's a lot of people that get diagnosed as stage 3, but in reality, it's stage 4 anyway. It just hasn't been detected as stage 4. And then certainly stage 4, which is metastatic, is definitely advanced or late stage for sure. Staging never goes backwards. So even if you become no evidence of disease, if you were stage 3, you're still stage 3. If you were stage 4, you're still stage 4. And so this is a great little diagram here. You can see these arrows all go from 1 to 2, 2 to 3, but you can't go backwards back to 2. And again, you can look at this more closely later on, and it defines the stages a bit more accurately. And there's actually a very complicated formula for determining whether you're stage 1 or 2 or 3 or 4, but we let the experts figure out that. So monitoring for progression. Firstly, we can look for hints without scans. So you heard Ellie talk about tumour markers, and I'm going to talk about that a little bit more in a minute. This is a chart where I've been getting my tumour markers measured for the last several years. And again, one of the things that's really important with tumour markers is to not get hung up about all the little ups and downs. If there was to be a trend that took me totally off the top of the chart, then you'd start to get worried. But when it's just going up and down like this, even if you see a trend twice in a row, it's too early to get panicky. Don't worry about it. But it's really helpful to me to make charts rather than just looking at a jumble of numbers. So I encourage people to do that, and it makes it a lot clearer. You can also do liquid biopsies that look at tumour cell DNA in the blood, which is known as ctDNA. And there's a number of different tests out there: Signatera, Guardant 360, Gallery, and there's going to be a lot more of them coming. Liquid biopsies is an incredibly rapidly advancing field, and it wouldn't surprise me in a few years' time if it actually becomes the gold standard ahead of tissue biopsies, which is what we use at the moment. And then lastly, as a hint as to whether you might be getting progression, is if you notice new or worsening signs or symptoms. It doesn't mean for sure that there's progression. There can be many other reasons that the signs or symptoms change or worsen, but at least it's a hint that it could be happening. But definitively, we need to look at scans, and it can be either a CT scan, sometimes known as a CAT scan, stands for computerized tomography, and also it can be from a PET scan. And PET scans, you can chart what's called a standardized uptake value, or SUV. And again, I'm going to use my own personal example here. This was my SUV at 25 when I was first diagnosed. And then by the time the tyrosine kinase inhibitors started kicking in, the activity went way, way down. And then you can see it started creeping back up, and we knew something was not going right. So if I hadn't been getting a PET scan done, we wouldn't have known this was happening. And we would have had to wait for the tumour to actually start growing or for it to spread somewhere else. But because I had regular PET scans being done, I was able to see that something was going astray, even though the tumour was not growing in size, and even though I was feeling totally fine. And so we actually did radiation, local consolidation therapy at this point, and you can see it brought the SUV all the way back down again. And so far it's been staying pretty stable. That's been 2.5 years now. So it really worked fantastically well for me. And then also CAT scans and PET scans don't work in the brain. And so we get what's called a brain MRI. And there's a few people, I think mostly outside the US, that seem to have doctors that aren't aware that our lung cancer can spread very easily to the brain. And getting a regular brain MRI is super, super, super important. Ellie talked a little bit about frequency, 3 to 6 months for people that have had brain mets already in the past, 6 to 12 months for people that have never had any brain mets in the past would be probably appropriate. Results can vary a little bit with scans. As I said, the technology keeps changing. None of them are perfect at detecting cancer, and they're read by people, and people make mistakes. Radiologist is the person that reads scans, and I'm here to tell you, just like any other human being, they can skip over things and not see something right. So it's really good to have an oncologist who also looks at the scans, because they may see something that the radiologist didn't, or they can at least confirm that what the radiologist wrote on the report, that they agree with it. So I do encourage you to have an oncologist that does look at the scans. So just briefly about what we colloquially call them, the Camage tumor markers. Dr. Ross Camage at University of Colorado published a paper, I think it was 2017. And again, you'll be able to click on this link when you look at the PDF. But he follows 4 cancer markers. And so sometimes people ask, which cancer markers should I follow? These are the 4. And why would you do this? Why would you bother going to the trouble and worry of doing cancer markers? Basically, if we can catch the tumors before they progress, or before they progress too much, then we can do something that is going to be a simpler treatment. So oligoprogressive, and we're going to talk about this as a new word later too, but oligoprogressive means that it's only gone to a few sites. It hasn't gone crazy all around the body. And if you're in an oligoprogressive state, you can still do localized treatment rather than having to change the systemic treatment. And that's really important, because we don't have lots of options for systemic treatments. So trying to catch a spread early is really important. So we want to look at the location of the metastases. It might be in the body only, or it might be in the brain only, or it could be in both. Or it could be what's known as leptomeningeal disease, and some of you would be familiar with this term. And that's when the cancer is spread to the fluid that surrounds our brain and spinal cord. And then we also want to look at the number of sites. So oligoprogression, that term I just mentioned a minute ago, the current definition for that is if there's 5 or fewer metastatic sites. Now, again, there's probably a little bit of leeway there. If you had 6 sites, it can probably still be treated as oligoprogression, but that would certainly be up to the doctor. But if it's gone crazy and it's all through the body, that's known as systemic progression. And this little diagram will give you a pretty good idea. So this is localized, it's just in the one area in the lung. These 2 are oligometastatic, and then this is where it's gone crazy and spread everywhere, and that's called widely spread or systemic progression. Oh, sorry. And then the question will be, when will I get progression? And the answer is we really never know. I used to be of the mindset, well, the median time that alectinib works is a little under 3 years. And gosh, I'm at 4 years. It seems like things are even more likely to get progression now because I'm past the average. But actually, Ellie did a very elegant study or analysis of a study that showed that the likelihood of progression actually becomes less every year that goes by. And she ran that analysis by Dr. Camage, and Dr. Camage agreed with it totally and said that it was totally in line with his own personal experience. And so if you can make it through that first 2 or 3 years, the likelihood of progression actually becomes a little bit less every year that goes by. When will you get progression? Again, I was able to see that I was about to get progression by following my SUV values on my PET scan. So that's another way of maybe being able to get a little bit of a clue of when progression might be about to happen. It's really important to be able to differentiate between progression and things that look like progression but aren't progression. So those of you that follow the support group page would be quite aware that Ellie has become a real expert on radiation necrosis because her husband had that problem. And it can be very, very, very difficult, even for the leading experts, to be able to differentiate between a brain met and radiation necrosis. But it's really important to have your doctor be very sure which it is, because obviously the treatment is going to be absolutely different depending on whether it actually is progression with a brain met or whether it's radiation necrosis. Similarly, is it really a new area of cancer in the lung, or is it a lung nodule? And again, that can be very, very hard to differentiate, and sometimes just requires a watch and wait attitude, because sometimes the experts just can't tell the difference. So once we think that there is progression, then we need to get a biopsy, because we need to understand what is driving the progression. Sorry, I just need to watch the time here. And so there's different types of biopsy. One is called a tissue biopsy. And the tissue biopsy can then either have a next generation sequencing or NGS analysis done, or it can have a FISH analysis done, or it can have an IHC analysis done. Won't get into the differences. You can get a liquid biopsy. And again, there's different companies and they're all a little bit different, but they're all NGS, I believe. And what's really important is for liquid biopsies, if you get a negative result, it doesn't mean that there is not progression. But if it is positive, it does mean that there definitely is progression, and they should be able to get some useful information from that. A common question is, which sort should I get? And the answer is, if you can get both, I recommend, and the doctors, leading doctors recommend getting both the tissue and liquid biopsies done. For those few that get leptomeningeal disease, there is a different type of biopsy that can be done for that. And again, I won't go into details here, but it'll be on the PDF for people to refer to later. Very important thing to understand is that tumours are heterogeneous. Heterogeneous means not all the same, not uniform, not constant. And tumours are not constant, not the same. They're heterogeneous, both between tumours. So if you've got a tumour in your bone and a tumour in your lung, or two different tumours in your lung, they may not be exactly the same. And similarly, even within a tumour, they're not exactly the same. And this is really important to understand, because when you get a biopsy with a tissue biopsy, it's just doing a sample, and that sample might show something that misses out on the area that's actually making the cancer grow. And that's why getting a liquid biopsy as well is a really good thing to do, as well as the tissue biopsy. There's pros and cons with each, and so getting both is the best thing to do. The results of those biopsies will help to guide the treatment that you're going to get. And we use what are called IC50 resistance tables, and we're going to talk about those briefly. So resistance is the cause of progression. The cancer cells mutate, and then the drug no longer stops the cancer from growing because the cancer cell has become resistant to that drug. There's different types of resistance. One is called on-target resistance. And they can be a single mutation, and they have these complicated combinations of letters and numbers that look like this. Or they can be compound mutations. This tends to happen after you've already had one TKI and then you go on to a second one. Or it can be not a ALK mutation at all. It can be something that's not ALK. It can be something like EGFR or HER or MET or RET. Or it can be even something where it's not even adenocarcinoma anymore. It becomes something totally different. It might go from adenocarcinoma to squamous, or it might go from adenocarcinoma to small cell carcinoma. And it's important to understand and know this, because this is going to determine what treatment is going to be the right treatment for you. There's no point just going to lorlatinib if you've got an off-target resistance. There's no point going to lorlatinib if you've got small cell carcinoma. So you need to know, why did the treatment stop working? And is my old treatment still working at all? And the answer usually is yes. So the experts are generally recommending, even if you've got off-target resistance or changed histology, still stay on the ALK TKI, because it's still controlling a lot of the cancer. So a recurrence is where the cancer comes back in the area where the treatment didn't eliminate everything, and you're left with persister cells. And then over time, these persister cells grow into tumors or cancer that you can detect. You can get local recurrence where it's in the same place, or you can get regional recurrence where it goes to the lymph nodes or tissues nearby, or you can get distant recurrence where it spreads to a remote or distant site. So this is a good little diagram. So primary resistance and acquired resistance. These are two terms that you want to learn about. With primary resistance, what happens is that you take the drug, but the tumor already has some cells in it that are not susceptible to that drug. So you can see in this second picture, all the gold colored cells have all died, but now you're left with the red ones, and they're resistant and they start multiplying. So that's primary resistance. Acquired resistance, you take the drug, it works, it shrinks, but there's still some persister cells, and then one of those persister cells changes and eventually becomes resistant, and then it starts growing, and now you've got a resistant tumor. So primary resistance, acquired resistance. So I mentioned earlier about tumors being heterogeneous, so you can see that in these diagrams here. And as I mentioned earlier, there's a heck of a lot of cancer cells in even a tumor that's just 1 centimeter diameter, and even something that's just 1 millimeter, which would never get picked up on any sort of scan. It's about 1 million cells there. So this is a really complex diagram. Anyone that doesn't know what's going on would look at this and say, oh, I give up. I can't understand that. That's too crazy. I don't know what that's about. This was presented by Dr. Schneider. She did an ALK talk for us about a month ago. And this is widely referenced by other doctors. It's really brilliant. But you look at it and it's like, I don't understand it. But what about if we simplify it? What about if we just take one part of that picture and try to understand that? Well, now we can. Now we've got on-target resistance. We take an ALK TKI and it works on the cancer. Let's make it even simpler. Let's highlight the treatment, the green box. Let's highlight the resistance with a red circle. Okay, so now we go to the next part of that picture. Here we've got a first or second generation drug. It's not working anymore because there's a new mutation. So now we're going to treat it with lorlatinib. And here's all these different types of mutations. As I said, they've got these complicated numbers and letters. So what about when alectinib stops working? So, well, here's our third generation drug, alectinib. It didn't work anymore because we've got a compound mutation, but it is still on target. It is still an ALK resistance. And so that's where these fourth generation drugs that are in trial are hopefully going to be really helpful, is for these compound ALK mutations. But then there's off-target resistance. So it can be MET or SHP2 or MEK or all sorts of other things. And these are bypass pathway activations. And so there's inhibitors that you can take for SHP2, and there's inhibitors that you can take for MET, and there's inhibitors that you can take for MEK. So there are treatments that can be done for these off-target resistance mechanisms. Or what about if it changed to squamous or small cell? That's called lineage transformation. And so now we see this whole picture, and suddenly it's not so complicated. We can understand it. So this is learning how to learn. So we're getting very close to the end here. Thanks for bearing with me. This is what's called an IC resistance table. Again, many of you have seen this on the support group page. And on the one on the left here, we've got all the different ALK TKIs, even all the way up to TPX0131 and Nuvalent 655. And then you've got the different types of mutations, and the red and green. Basically, green means it's working great, and red means it doesn't work at all. And similarly, we've got another table here which uses, even includes gilteritinib, which is being about to go into trial at the University of Michigan. So you can see all these different things here. And so just wanted to help you understand what these tables mean and how they work. And again, these will be on the PDF for you to look at later on. So you need to understand these, and similar tables are based on laboratory experiments, not patient studies. But there have been studies showing that they seem to, basically what we're seeing in the lab does seem to play out in real life as well. And they definitely can help to guide the treatment of which TKI you should take next. And that's why it's important to understand these tables and everything else I've been talking about up till now. So, treatment resistance. I love Hagar, it's one of my favorite cartoons. Overcoming your cancer's resistance to treatment requires understanding why it is resistant and then using the right tools to overcome the resistance. So that's the end of the presentation. Thank you, everyone. I'll be doing Q&A in a little bit here. Progression is part of the journey. Lack of progression is also part of the journey. If you haven't already done the surveys, please do. The researchers are going to learn huge amounts and improve their research. Dr. Culver's been doing an amazing job working with pharma to try to expand these studies even further. And again, please, as Mark mentioned earlier, become knowledgeable, watch the ALK talks, watch the past summits, which are also on the YouTube channel. Lots of great information. And the next session will be next week, and we'll be continuing what we started today and really learning what to do when you've got cancer. So thank you very much. Be well. People that know me know my favorite saying is treasure every day. And if you've got any questions, here we are. And lastly, as I always say, go research.
Well, great job. And I'm going to start firing some questions at you. Tumor growth level and the markers, the blood testing that you're talking about, the CEA markers, as patients, do we advocate for those?
I'm sorry, could you repeat the question?
How do we advocate for as patients to our oncologists who do not use blood tests?
The tumor markers, you mean?
Yeah.
Great question. So really, I think the best way to go about doing that would be to get a printout of that paper that I will give the link to in the recording, and then either print it out or email it to your doctor, whatever's going to work best where you are. And then when you meet with them, let them know why you're interested and say, here's the paper that documents it, because doctors generally don't want to listen to what a patient says. They want to listen to what other doctors said.
Many of us don't receive PET scans unless we had something that showed up on a CT scan. There's a question here: is how often were you getting PET scans? And you showed us your chart.
So it varied a lot. Really, I was getting them done all the time. I wasn't getting CT scans done at all. And then my oncologist shared with me that she had been bitten a couple of times where there'd been a change in the tumor size that hadn't shown up on the PET scan. So I had mentioned that PET scans and CT scans have their own pluses and minuses. So one of the pluses of a CT scan is that it actually detects change in size more accurately and more precisely than a PET scan. So there's pluses and minuses to each. And depending on the situation, I've been I'm pretty much at a stage now where I alternate unless things have been looking really shaky. I've been kind of a little bit shaky the last year or so. So I've been mostly doing PET scans the last year.
I don't think most people know that the liver is your baseline of your SUV, correct?
I'm not aware of that. Basically, what I know is that they kind of measure the SUV as a background. So there's always sort of some background noise, and then they compare the activity. So, liver might have a different level of activity to bone, to something else. Maybe Dr. Culver can enlighten us a little bit more on that one.
Ken, can you, I know on FCVs, we have different opinions that come through on Facebook. What is the baseline level that they go
Yeah,
off of?
I'm not qualified to answer that. If Matt Quirk is on, he's an expert in that. We should get his opinion. Mark, maybe we can then circulate that to everybody.
All right, sounds good.
Are you here,
Ken,
Matt?
I thought Matt was here, but maybe he dropped off. I don't know. How often do you get your blood markers, Colin?
I get my blood markers done every time I get a blood draw done. And again, that varies, but typically every 6 to 8 weeks.
I'm just curious, if I walked into Quest Diagnostics, could I get my blood markers?
They have to be ordered by the doctor.
Gotcha. All right. Just scrolling through here. Can you explain the IC50 values more?
Can I explain the IC50 values more? Basically, the IC50 values, and again, Ken will be able to explain this much better than I can, but they're an indication done in the lab. They get a dish with cancer cells. They sprinkle it with the drug. They don't really sprinkle it with the drug, but they apply the drug, and they see how long it takes for the or how much drug it takes to reduce the number of cancer cells by half. And
I think would be kind of cool. Dr. Lynn, I have a post here right here. It says, Dr. Lynn does not believe in value of tumor markers and believes they are anxiety provoking. So I think we should have a debate between Dr. Lynn and Dr. Gamage and an ALK talk.
It's probably already been done, I imagine. But
you think so?
Yeah.
So even actually, there's a very good presentation that's on the YouTube channel from the summit last year, and it's got Dr. Schenk, who works with Dr. Gamage. And Dr. Schenk does not believe in doing tumor markers. And they had a discussion in front of the camera about the pros and cons of it. So anyone can go and watch that discussion. It's quite entertaining, actually. And yes, absolutely. Again, that's something where patients, if the patient's going to get stressed out about it, they shouldn't do it.
Yeah.
That's basically the bottom line. If you're going to stress out about it, don't do it. if you're going to, for me personally, I find them kind of like scans. People think scans are anxiety inducing. To me, scans are the opposite. It's like, oh, great, I got my scan done. I can go and live my next 3 or 6 or 9 months. Fantastic. And it's like that with tumor markers. I get my tumor markers done at the 6 or 8 week mark, and everything's within normal range and not going crazy. It's like, yes, everything seems on track. I'm feeling good. The tumor markers look good. Things are going well. So for me, it's a reassurance, but it really does depend on the individual. And it comes back to what I said at the very beginning of the presentation, that it really depends on every patient being different. We've got to treat all patients as individuals.
I like how you explain NED. I've never dated NED. I've always been stable. So I have no idea what that means, NED. But is that like a false sense of security? Do you think that people feel like, oh, I'm NED, like I can maybe skip some of my pills? It's like,
right. Yeah. I mean, how it could be, and that's why I made a real point of saying that NED does not mean that you're cancer free. You could have multiple sites throughout the body that still had 500,000 cells in each of them. You could have millions of cancer cells still floating through your body, and it says that you're NED. So to me, yeah, just if you're feeling good and life's good, that's what matters.
that's
Hey,
a
Mark,
question for,
it's Kent.
yeah,
Can I add something on this?
absolutely.
Colin's description of IC50 is really important, and I want people to make sure they understand. IC50 is a laboratory test. It's not being done on your cells. So if you look at this group of people who are all on Zoom, nobody's got an identical tumor. So the companies take a few standard cell lines, they test them across all the agents. As Colin said, they do increasing concentrations. IC50 is the C stands for concentration. 50 means where 50% of the cells are killed, not 100%, 50%. So it's really a good guide, right, for about which drugs work on which mutations, but it doesn't necessarily mean that things won't work for you, because this isn't being tested in your cancer cells. And one of the nice things that we're doing with the University of Michigan is they're specifically trying to ask that question of which drugs will work on your tumor. So it's just something to keep in mind. And as all the things Colin said, all the numbers are, whether it's PR or CR, what's the likelihood, are based on a population. They're not based on you. And so we have, so it's just important to keep in mind that those are really good guides, but they're not definitive, just like the tumor markers that Colin's bringing up. They're not definitive, or there'd be no debate. They're an adjunct that some physicians find helpful, some patients find helpful, but I would imagine it's the minority of oncologists to use them because they have not yet been validated in a clinical trial.
As an oncologist, I want to ask you a question, and I don't want to blow up Colin's magnificent presentation, but is too much knowledge get in the way sometimes of a patient to a doctor relationship?
Well, that depends a lot on the personality. And we've heard examples where, doctors won't even allow, are not interested in patients that get a second opinion.
Mm-hmm.
Right? That's one extreme. And on the other extreme, it's someone like what Kirk Smith said, his doctor encouraged him. So you're going to find that on knowledge too. Some are going to really love it, that they can have a meaningful discussion with the patient. And there may be those who find it really uncomfortable, maybe even threatening, especially in the time that oncologists are given to be with a patient. That impacts a lot on their patients, for patient CE for time with patients to have those discussions, Mark.
I think Colin should open up a second opinion program.
I think he'd be good,
and
really
Zoom in
great.
with us.
Yeah, yeah. That would be great, Colin. You need to get your insurance certifications back.
Yeah.
I can give second opinions about dentistry, but that's about it.
There's a lot of talk in our groups about variants. I know Dr. Lamb was asked about the variants in our last summit, and his basic comment was, we really don't know yet about the treatment of those variants. So anyone want to comment on that?
Well, I will tell you, there's a lot of discussion around the meaning of the variants, and the people I've met in the ALK community, we have people who are variant 3 who have been on alectinib for years and have no evidence of tumor disease.
That's
So
me.
again, each person, yeah, there's 2 now, I know. So they're a guide, but there's more to the ALK positive tumor than the variant. And it could be P53 status, which is a tumor suppressor gene that can be inactivated in these cells, could be way more important than the variant.
And it can
And
be even more than that. So I am P53 and I'm variant 3 and I'm squamous. You'd think I've got everything stacked against me.
Mhm.
Here I am 7 years later.
Yeah.
I think it's your accent that's keeping you in the game. The other thing too is I want to take something that Jeff said in his and bring it forward to our next conversation, which is you might have to get in your mind that you will progress. I thought when I got crizotinib, I'm going to be living forever. I'm crizotinib. 8 months, PFS, boom, I'm out. So, Colin, what's your opinion? You brought it up a little bit in your slide.
So, I mean, maybe there's going to be 1 or 2 patients out there that die of something else before they die of, ALK cancer. And this is why I had an interview in person with Dr. Camage about 3 years ago. And he said, Colin, my wish for you is that you die of something else before you die of cancer. In other words, go and live your life, go enjoy your life, go traveling, go sailing, go hiking, go do whatever you like doing, live your life. Yes, progression is going to come along at some point. You don't know when. And so just get on and live life. And like I said, back in 2016, we didn't, thinking 7 years was like, it just wasn't a thought. It just never crossed your mind. And the idea of going out and buying a home, it was, it makes decisions in life very, very, very difficult. Mark just got married. And, so you just got to get on with life. But yes, progression will happen. And as Jeff said so well last time, get yourself prepared, get everything organized, and then keep yourself up to date, but get on with life. Don't dwell on the fact that it's going to happen because you don't know when.
Is there any homework we need to do before next Sunday?
I'm sorry?
It's a question I'm giving you.
Oh.
Is there any homework that's assigned to us that we need to bring back for next Sunday?
No, I think just, maybe rewatch this video, rewatch the ones that Ellie and Jeff did, and come along next Sunday.
I think the most important thing that people need to do is to pass around the knowledge that these videos are being made. Mentees, mentors, whatever, we're making this into a whole collection. And I really, really appreciate the PowerPoint. I thought you did an unbelievable job. And thanks so much. So.
Well, thank you for the opportunity.
Yeah. You guys gonna mute and say hello to Colin, yes.
Thank you, Colin.
Thank you, Colin. Great job.
Thank you,
Thank you,
Colin.
Colin.
Thank you.
Thank you.
Thank you.
Thanks, Colin.
Colin.
Congrats,
Thank you,
Mark.
Colin.
Thanks, Colin.
Thanks,
Thanks,
Colin.
Colin.
Thank you.
And Summer.
Thanks.
And Ken.
Yep, thank you.
All right, guys, we'll see you next Sunday.
Thanks, Mark.
And girls.
Thank you.
Thank
Thanks,
you.
Colin.
Thank you.